For the next few minutes, I'd like to focus on volumetric analysis of structural MR images, specifically looking at some of the core biomarker regions typically employed in clinical trials, so primarily the hippocampus, but also the whole brain and lateral ventricles. Volume measures and volume change measures in these regions have been assessed with several established methods within the literature and in previous trials, such as the BSI, Sienna, and FreeSurfer pipelines. These methods have demonstrated good sensitivity and accuracy and reproducibility, but may not necessarily have the flexibility to adapt well to new regions of interest, and can sometimes require manual intervention in order to achieve optimal results. And this may take the form of semi-automated initialization, parameter tuning, or post-processing and segmentation refinements. At IXICO, we employ fully automatic multi-atlas based approach called LEAP, which we've successfully deployed in several clinical trials over different therapeutic areas. In this pipeline, in brief, T1 images are pre-processed, skull stripped, and in homogeneity corrected. Input images are then compared to a reference database of T1 images with pre-existing segmentations for the region of interest or the parcellation schema. These segmentations for the most similar database images to the input selected in a manifold space are transformed to the input image, a majority voted to create a consensus initial segmentation, which is then automatically refined. This provides robust, fully automatic cross-sectional segmentations, either of individual regions or whole brain parcellations without the need for manual intervention. Volume changes can be estimated with a subsequent follow-up pipeline, which employs 4D graph cuts. And in this approach, regions of interest are simultaneously segmented in a follow-up and baseline image, with segmentations being temporally coupled and initialized by the baseline cross-sectional segmentation. And this provides us a way of measuring volume difference between these simultaneous segmentations, which we can then use as a volumetric endpoint. So this approach has the advantage of being both fully automatic, so there's no manual intervention involved, but also very flexible, allowing us to update the reference database that generates the consensus segmentation to include subjects from different therapeutic areas or different populations to best fit the study under investigation. We can also easily update the parcellation schema to include new regions of interest that are disease relevant. And this is what you see here, an example of the neuromorphometrics whole brain parcellation schema being updated for progressive supranuclear palsy in the brainstem. This approach can also be modified to segment individual regions of interest for highly optimized volumetric estimation. And this is exactly the approach we've applied to the core AD regions of interest, so the hippocampus, whole brain, and lateral ventricles. Starting with the hippocampus, we validated a implementation of LEAP against a gold standard consensus-based hippocampal data set. And in this protocol, 2 expert raters manually segmented the hippocampus for 135 subjects. And these were merged to create a gold standard reference data set, which we used to validate the pipeline. We see very good spatial overlaps, as shown by the dice scores in the lower left, as well as good agreements in absolute volumes and significant correlations. Notably also, the variability between the manual and the automatic segmentations is in line with inter-rater variability. The variability, in addition to direct comparison of volumes, we also validated LEAP in terms of accuracy in predicting 2-year conversion from MCI to AD diagnosis using empirically derived threshold. Also considered were the MTA scores and lateral ventricle volumes as predictors for conversions. And hippocampal volume was found to be one of the most sensitive predictors to conversions, with the LEAP approach providing comparable accuracy for prediction of conversion within 2 years to manual segmentations. And details of that can be found in the paper reference there. In terms of volume change, which is a key biomarker in many trials, we validated the LEAP pipeline against manually initialized or semi-automatically initialized BSI measures and free surfer for a 2-year decline in 100 subjects. We compared the pipelines in terms of sample sizes needed to detect a 25% change in atrophy values for a hypothetical 2-arm clinical trial. And we see that BSI and LEAP perform significantly better than the free surfer pipeline, as shown in the table, specifically the last column showing sample sizes, with LEAP performing only marginally worse than the BSI. However, it's worth noting that LEAP is fully automatic and flexible, whereas the BSI was manually initialized and potentially susceptible to vulnerabilities of intra and intra subject variability for those initialization segmentations. Also being fully automatic, it has much reduced resource cost for deployment, meaning much faster turnaround times in terms of volumes. For the whole brain, we did a similar validation comparing the LEAP implementation against semi-automatically initialized BSI, again showing comparable results with the BSI with significantly correlated volume estimates and sample sizes, again necessary to detect 25% change in atrophy, as shown on the right, as well as sensitivity to predict clinical groups from volume alone, as shown in the lower table. And very quickly as well, lateral ventricles were also validated with a very similar approach. Again, semi-automatically initialized BSI was used as a reference method, and we see that LEAP performs comparably in estimating absolute volume changes, but also in terms of group separation, where there was no significant difference between the methods. So in these slides, we've demonstrated that the LEAP method performs comparably against a commonly employed standard, so the BSI using best practice semi-automatic initialization. However, LEAP has the advantage of being fully automatic, flexible, and scalable to allow therapeutic specific validation in clinical trials. In the next few slides, now that we've seen the tools, I'd like to discuss some of the applications of these, specifically with the examples of using hippocampal volume in efficacy analysis and patient selection or stratification and sub-analysis. So in order to assess treatment efficacy, historically clinical rater scales have been used as an endpoint. So whilst cheap and simple to administer, they are very coarsely granular and potentially insensitive to subtle effects. However, quantitative volumetrics provide a much finer granularity, which shows significantly greater sensitivity to efficacy, as demonstrated here in a table comparing ADAS-Cog, MMSE, and hippocampal volume and whole brain necessary sample sizes necessary to detect, again, a 25% change in a 2-year clinical trial, and details can be found in the paper linked in the bottom corner. Really demonstrating the use of quantitative measures for reducing sample sizes, and this has obvious downstream implications for patient recruitment feasibility and clinical trial costs. Also, as mentioned in a previous slide, hippocampal volume has been shown to be a sensitive predictor of cognitive decline. Specifically, the normalized cross-sectional hippocampal volume can be used as a feature to assess whether a subject will likely convert from from an MCI to an AD diagnosis within a 2-year period, using empirically defined thresholds from independent data. In this example on the right, thresholds were estimated from an ADLI data set. This finding is robust across multiple methodologies, but with LEAP being one of the leading 4 methods assessed in the paper on the right. So this indicates that hippocampal volume is a good candidate for enrichment, using it to identify patients with likely short-term decline for inclusion in clinical trials and getting potentially much greater power for efficacy and placebo analysis. So here on this slide, we show the impact of employing such enrichment in a clinical trial setting. We compare the effect of enriching with hippocampal volume to using amyloid positivity as a screening criteria. The graphs on the lower half of the slide show for an MMSE and ADAS-Cog outcome measure in a hypothetical 2-year clinical trial, the sample size is needed to measure a 25% effect on the right. And on the left, we see the SNR, which is the signal to noise, which is a measure of the 2-year change in the outcome measure divided by the standard deviation, and it informs the power of the signal of our interest. And overlaid on both plots, you see a line in blue, which is the expected screen failure rate. So moving along the bars from left to right, we have our reference unenriched population with associated SNR and sample sizes. By enriching with low hippocampal volume, so subjects with a normalized hippocampal volume less than a data-driven threshold, which put them at risk of likely short-term neurodegenerative decline, we see improved SNR and sample sizes, as shown by this N+ bar group. Another commonly employed enrichment criteria in more recent trials is to screen or perform stratified analysis based on amyloid positivity. And this has an even greater effect on improving SNR and reducing sample sizes for clinical trials. What's important to note, however, is that this neurodegenerative enriched population is not necessarily the same as this amyloid enriched population, which allows us to combine both screening criteria to create a amyloid positive group with likely short-term neurodegeneration. And this can result in a greater than 50% improvement in the signal to noise ratio and sample sizes compared to an unenriched population. So you may have noted that this also results in a expected screen failure rate of around 45%. However, due to the increased power of this enriched population, you wouldn't necessarily need to screen more subjects to power your clinical trial, as shown on the left. And in fact, you would enroll fewer patients in a longitudinal trial, and therefore, over the course of 2 years, your trial costs would be significantly reduced compared to screening on amyloid positivity alone. And again, further details can be found in the papers cited in the corner. So finally, my last 2 slides, no talk regarding MR volumetrics would be complete without at least mentioning AI, or more specifically, deep learning. There's been a strong recurring trend in the field to employ deep learning methodologies to neuroimaging challenges, in this case, segmentation. These methods have demonstrated great promise in volumetric analysis, and at IXICO we've seen that the use of a UNet architecture with data augmentation can provide robust regional segmentations across a range of scanner protocols for subcortical structures, for instance. This has been presented at several conferences, as shown in the bottom left. We're also currently working on applying and validating these methodologies for specific therapeutics, including the core Alzheimer's regions of interest, such as the hippocampus, whole brain, and lateral ventricles, where we expect and indeed are seeing increased accuracy and sensitivity for efficacy. And we look forward to reporting the outcomes of this work in the future as it develops.